So I'm going to talk to you today a little bit about biomaterials for natural polysaccharides, especially from agri-industrial and agricultural wastes. Some sort of way to give them an added value. But before I talk about our research here, I want to tell you a little bit about our university, for those of you who don't know. So I'm from Universidad San Francisco de Quito. This is a private institution. It's not a large university. It's actually a small university here in Ecuador. Yeah, as you can see here, we take very good care of our campus. It's actually being named one of the 10 most beautiful campuses in Latin America. Also, according to the QS rankings, we have been ranked as the best university in Ecuador and one of the top in Latin America. And this has been in the last at least 4 years that I've been at the university. We've been always at the top. And we are a liberal arts university, which means that our students not only learn about chemical engineering in this case, but they also learn about arts, about sports, and philosophy and other things that make them well-rounded individuals. Our Department of Chemical Engineering belongs to the College of Sciences and Engineering, which is the largest college in the university. And this is a very rare case for a private institution in Ecuador. So it is a great achievement for us. So the Poly, as we call it, has 11 engineering programs, only two sciences programs, which are math and physics. And the Department of Chemical Engineering is the third largest of the Poly. We have 13 professors with PhD and about 10 more, which are assisting these 13 professors. We have over 200 students. This is a very approximate number because I don't know what it is exactly right now. We have 8 teaching labs and 8 research labs. Some of them are actually quite new. So the university has been investing a lot in research. Within the department, there are 3 important research groups. IDEMA, which is the one that I'm part of, and I'm going to talk to you a little bit about it right now. There's also a computational and theoretical chemistry group, and applied sciences group, which they work more with the environmental side. So here's a picture of our department. Every year we organized chemistry Olympiads to give scholarships to high school students to study chemical engineering at the university. And this is the group picture that we took like last year during this event, obviously pre-pandemic. IDEMA, which is the Institute for the Development of Alternative Energies and Materials, which is the one I belong to, has 3 main research lines. One, which is biomaterials, which is the one that I lead, where we develop hydrogels, absorbents, or anything that's biomedical applications related to biomedical applications or biotechnology, especially agricultural biotechnology. We also have been doing some biodegradable fabrics, but that's a very new area. We also have a bioprocesses line where I'm part of also. We do anaerobic digestion, bioethanol production, and new technologies for the production of organic acids, such as citric and lactic acid. And then we also have a thermochemical line where we do mainly 2 things, cracking and pyrolysis. The cracking of used oils and plastics, and pyrolysis mainly of a lignocellulosic biomass, though we have been doing other materials recently. So this is basically brief information about the university, our department, and our institute. Now I'm going to talk to you about the biomedical side of biomaterials that we have been working on. And for those of you who don't know much about biomedical applications, these are actually quite diverse. But here I give you some examples: contact lenses, wound dressings, controlled delivery, tissue engineering. And this last one was my original main area of research, area of focus. This is where I did my PhD and my postdoc. But basically, for those of you who don't know, tissue engineering is about creating some sort of intelligent implants. And it has traditionally three main components, one being a scaffolding material, which is where the biomaterial part comes in, a cell component, a cellular component. This could be, stem cells, it could be specific cells from a tissue, what we call differentiated cells, and a molecular component that's basically growth and differentiation factors that will do some sort of tackle cellular activities of interest. Now, okay. Within these scaffolding materials, within biomaterials, we have been working a lot on hydrogels. And hydrogels are basically hydrophilic, three-dimensional polymeric networks that are able to absorb large amounts of water or biological fluids, for instance, and they do resemble, to some extent, biological tissue. And that's why it's so important for us to work on them. Now, this isn't developing hydrogels is an area of research that has been around for a while, but it doesn't lose importance because of their wide range of applications, wide range of materials that you can use to create them. And the main challenge that we face here in Ecuador is what materials, what polymers are we going to use to make hydrogels? And if you look at some of the examples of biopolymers that have been most widely used in the literature for this, I give you here only 4 examples, but there are many more. Collagen, especially collagen type 1, sodium alginate, polyvinyl alcohol, polyethylene glycol, which are, they have great properties. They also have their drawbacks. But for us, the problem is, how do we find that here? And you need to work a lot with thinking about the reality of the place where you are at. And so collagen type 1 is not found here. You have to import it. Same thing with sodium alginate and PVA and PEG. And these are very expensive materials for us here, especially when they're pharmaceutical grade. And so you have 2 choices. The first one is, well, you could produce them here. You could extract collagen from some sources or synthesize PVA and polyethylene glycol. Sodium alginate is a little bit more complicated because the algae where it is extracted from are considered invasive species here in Ecuador, so there's not even a possibility of cultivating them here. So we have chosen the route of finding alternatives. And Ecuador is considered a mega diverse country. It's one of the smallest but most diverse countries in the world because of the geographical conditions, I guess, climate conditions. And this is what we started to look at because it is so diverse. There's a great variety of things that are cultivated here. It relies heavily on agriculture, and so it produces a lot of agri waste. And in many cases, nothing is done with this agri waste. And we decided to look at this agri waste and see what we can find there. So I'm gonna take you a little bit through Ecuador. This is not a tourism ad, but probably will help you look at how beautiful the country is and how varied it is. First, we're going to go to the coast. So we have the Pacific Ocean right there, and we have beautiful beaches. This is one of my favorite places. It's called Los Frailes. But besides the beautiful beaches, there's also a huge area of shrimp. Ecuador is the second largest exporter of shrimp in the world. And because we produce so much shrimp, we also produce a lot of waste from shrimp. And there's nothing that is done with this. I mean, some things are done, especially animal feed, but there's a lot of waste still that is not used. And if you look at shrimp, you will find a very popular biopolymer, which is chitosan. Well, you find chitin and then you do deacetylation and produce chitosan. So here we have a very important source of a biopolymer that has a lot of potential because it's, it's biocompatible. It has fungistatic and bacteriostatic properties, and so it has been used actually quite a lot. Then, if we go now to the mountains, central part of Ecuador, the Andes, we actually have here the Chimborazo, which is a volcano, is the well, I don't remember the definition, but is the point in Earth that is farthest from the center of the planet or something like that. But anyway, very beautiful. So, but if you go to the Andes, now you find tubers. There's a huge variety of tubers, especially potatoes. I don't know how many types of potatoes, but it is over 50 types of potatoes that are cultivated in Ecuador, some more than others, but there are also other tubers such as sweet potato, which are also, there are also several varieties here, and cassava, which we call yuca. And what do these tubers have? They have a lot of starch. And starch has two very interesting molecules, which are amylose and amylopectin, that also have very interesting biological properties that we can take advantage of. Then, if we go to different parts of Ecuador, you can go to the coast, or here in the mountains, or if you go to the Amazon jungle, you can see here, you will find this very loved or very beloved fruit, which is cacao or cocoa, some people call it, where we get chocolate from. But all that that you see, all that outer layer of this cacao is a waste. And Ecuador produces about 200,000 tons of this waste because it is one of the largest producers of fine aroma cacao, which is the finest of the finest that you can find. And this waste has also two very important, among others, these two very important molecules, which are cellulose and lignin. And these are also molecules that we can take advantage of, and we have been working with them. And if you look at the entire country, as I told you, this is a mega diverse country, which means there's a great diversity in fruits, which are producing very large amounts and also generate a lot of waste. And that waste is very rich in this polysaccharide, especially polygalacturonic acid, which we call pectin. And one of the main focuses lately in our lab has been isolating, extracting, and purifying and characterizing pectins from different fruits. We have characterized over 12 fruits under pectins, and we have found very important differences in molecular weight and other things that have an important effect on, as I will tell you later, hydrogels. So now we need to look at these molecules, but the problem is generally, in some cases, we can use them as if, you isolate and purify them and you can produce hydrogels from them. But for example, starch, if you use it in its native form, it has a lot of drawbacks. It's not very stable in aqueous media, which is a problem for us. It doesn't have great mechanical properties either, so you have to modify them in order to create feasible technologies. In the case of chitosan, for example, which is the first polymer that we worked with, chitosan has one main problem, and it is that it's soluble in acidic pHs. And this is okay for some technologies, but if you want to create something that's implantable or autocrosslinkable that you could put in the body directly, that would be a problem. So we decided to make these modifications. One is putting them, creating a chitosan lactide, and the lactide will make it soluble at physiological pH, which is between 7.2 and 7.4. And also we put these azide groups, that make them cross-linkable under UV light. So if you have a solution of this modified chitosan under UV light, it would create a hydrogel. Now, In this case, we didn't create a hydrogel directly, but we used a hydrogel-like coating for metallic implants, especially titanium, a titanium alloy, which is the most widely used alloy for metallic implants. And we created these coatings on these titanium implants, and we encapsulate in a very important protein that induces differentiation of stem cells into bone cells, what we call osteoblasts. And we cultivated these mesenchymal stem cells, cells from bone marrow, and they underwent differentiation that we could see through different markers. And one of the most obvious ones here is the formation of these calcium deposits here that we characterized and confirmed that these were calcium deposits. Now, using the same idea, we also created hydrogels with hydroxyapatite. Hydroxyapatite is a mineral component of bone, is an analog of the mineral component of bone. And we also cultured mesenchymal stem cells on them, and we followed their differentiation. The idea here was to create some kind of bone fitting for maxillofacial applications, so that we could have, something that's injectable, for example, a jaw defect or something like that. Then we started working with chitosan and starch, and this was very interesting because I met this researcher who worked with starches. She does all starches for food applications, food science. And she said, I have this methylated starch that doesn't work very well for our applications, but when it dries, it forms this very rigid plastic. So I said, well, let's combine it with chitosan and see if we can do something about that. The problem initially was that the plastic was very soluble in water, and this is why we incorporated chitosan. So we created these hydrogels with chitosan, and I'm not gonna go into detail here, but we did some metabolic activity tests on dermal fibroblasts, which are skin cells, and we demonstrated that these were cytocompatible, which means, they're not gonna cause cell death or cell damage. Actually, cells proliferated on the material. But the problem with this technology was that in order to create these hydrogels, we had to use a cross-linking material that's called glutaraldehyde. And glutaraldehyde is highly toxic, and it was a huge problem for us to get rid of the glutaraldehyde. We did it, but it was a very complex process to get rid of unreacted glutaraldehyde. So we decided to look at, amylose and amylopectin structure, and we said, well, if we oxidize the structure, these primary alcohols will turn into carbonyl groups, and carbonyl groups from these amylose amylopectin modified molecules in starch, when they meet with amine groups from the chitosan, they could form a shift base. So ideally, if we met these two, put these two together, they would create cross-linking and we would be able to form hydrogels. So we went into modifying starches with sodium periodate, did all the characterization, and when we mixed this a suspension of oxidized starch and chitosan, we were able to form these hydrogels. We also, I have to say, we used here also this modification of chitosan lactide. So the idea here is you have a solution of chitosan with probably mesenchymal stem cells. You mix this with oxidized starch suspension, and then you should be able to inject it, for example, in an arthritic site and promote cartilage regeneration. We were able here to control the time of gelation, which means we had hydrogels that would gelate in 2 minutes, but also some that would gelate in 10 to 15 minutes, so you would have time to inject them. Please let me know how I am in time, because I can get carried on, and please let me know if I'm okay with time.
Time is good. Sorry about that.
All right, thanks. So we did a lot of research on, the oxidation degree, the composition of the hydrogels. We found that the hydrogels were indeed cytocompatible. However, when we tried compatibility of the components of the hydrogels alone, the high oxidation degree starch caused cell death. So we decided that this was not suitable for biomedical applications, but low oxidation degrees and medium oxidation degrees work really well. Then we also worked, and this, well, this is not exactly chronological, but this is a recent paper, actually our most recent paper. We started looking at PVA. Why? Because PVA forms excellent hydrogels, great mechanical properties. They have very interesting properties in terms of drug delivery. But the problem is, to form PVA hydrogels that are stable, you need large amounts of PVA. In the literature, they use up to 35% weight percent of PVA, which would make them actually very expensive for us. So we said, what happened if we partially incorporate oxidized starch into the PVA hydrogels? Can we go down on the PVA concentration and still have stable hydrogels? We were able to go down to 10% PVA, in some cases 5% PVA, and form stable hydrogels. Now, what we did in this paper was look at the effect of different oxidation degrees. And we found that we encapsulated here ibuprofen as a model molecule, model drug. And then we look at the release of the ibuprofen from these hydrogels. And we found that there is a very important effect of the oxidation degree on the way the molecule is delivered. But also we found very interesting effects on swelling. So swelling is how much water they absorb, and the higher the oxidation degree, they were more stable, but the swelling was lower. So you kind of have to compromise a little bit, and it depends on the application that you want to give. So if you want to look into tissue engineering, you probably would be okay with something that doesn't swell as much. But for drug delivery, you probably need something that swells a little bit more, because you can see here, for a large oxidation degree, you get only up to 20-something% of the ibuprofen released. Then after that, we went and looked at the oxidation technology, oxidation method, but not this time with starch. We went and looked into pectin, which is this polygalacturonic acid that we find in fruits, especially fruit peels. We did the oxidation of pectin, and this time we used hydrogen peroxide, and we use a high oxidation and low oxidation also degrees of the pectins. Here you can see scanning electron micrographs of the hydrogels that we formed. In this case, these are oxidized pectin with gelatin. And we found very interesting porous structure. This low oxidation was particularly very interesting because they formed this very organized pattern. I mean, for hydrogel, these organized patterns are not that common. And we were able to reproduce this behavior. We still haven't found an explanation for this, but it's been published. And we found very interesting differences in swelling behavior. This curve that you see here, this is native pectin, hydrogels with native pectin, native pectin that hasn't been oxidized. And you see that the swelling starts decreasing, which means the hydrogels start to degrade. And we actually saw that in the lab. And the swelling was also controlled by how much of the oxidized pectin you put in. And then we looked at also cumulative release. In this case, we use acetaminophen, and we also saw very interesting differences in the release profiles. So you could tune the release profiles based on how much oxidized pectin you have, and also what type of pectin you're using. I didn't put these results here, but we started we did this with orange pectin. Then we started looking at different fruits, and I'm getting some messages here. Let me see if they're not. Okay. Then we started seeing different fruits, and we also saw very important results. This is being published now. I mean, it's under review, but in terms of time, I'm not showing that here. Then we looked at now pectin also, but now with chitosan. You know, gelatin has has some amine groups, but chitosan has a lot of amine groups, and we created these hydrogels with chitosan. You can see here the hydrogels are a little bit, to our surprise, they didn't look as compact as the ones with gelatin. However, they were way more stable. The gelatin pectin hydrogels were stable up to 32 degrees, which means I could not put it in the body. I could use it for transdermal delivery, like skin patches, but not for internal applications. These were stable at 37 degrees Celsius. And we also saw very important differences in swelling. I'm not going to go into much detail here. And in the release, we also saw a lot of, interestingly, when we have more pectin, they were able to swell less. And that also had an influence on the release of acetaminophen. In this case, we also used. Now, I told you a little bit about the hydrogels that we've been working on: pectin, chitosan, and starches. And starches are the polysaccharides that we have been using the most. But now I'm going to talk to you about something that is not hydrogels. These are actually porous sponges, and this is done with cacao, with cocoa. As I told you, Ecuador produces a lot of these residues, which are here are the seeds that are used for chocolate, and all of these are residues. So they're rich in cellulose and lignin and pectins, hemicellulose. And so we said, can we create porous structures from this material? And so we did different treatments. We did treatment with acetic acid, sodium hydroxide, different only water, different conditions in temperature and time. And we were able to find these porous structures, especially with the acidic and the alkaline treatments. With water, we saw some porosity, but we demonstrated later that this was superficial porosity. It wasn't internal porosity. And here you can see the untreated, which is very similar to the water treated sponges. Then of course, we did a lot of material characterization. I'm only showing you here some of the cell work. We demonstrated that the acid treated samples were not cytocompatible according to the norms that we used. So the alkaline treated ones were the most cytocompatible. So after that, we decided to look into more details on this treatment, on the sodium hydroxide treatment, and we created sponges with different compositions in terms of cellulose and lignin, and we saw very important differences in the thermal properties, temperature of maximum degradations and residues for the thermogravimetrical analysis. We also did some FTIR, and we saw the disappearance of peaks that are associated with lignin. So what the sodium hydroxide does is it swells and breaks this lignin structure, which gives more rigidity to the material and also lowers the capacity for biodegradation. And so our idea was to eliminate as much lignin as we could and keep some more of the cellulose. So we saw some of those peaks that are shown here with the red arrows disappear or diminish. And you can see here that also the porosity changed with different compositions. So we're still working with this because we also want to look at ways of the way we produce the sponges was a little bit manual. So now we're trying to look at how can we do this for mass production, for example. And one thing that was very interesting with these sponges is that they had the ability to swell up to 2,000% of their weight. So these are super absorbents that could also have applications in other areas, not only in biomedical, in the biomedical field. And we're also looking into that. So,
Excuse me, Dr. Alvarez.
Yes.
Sorry for interrupting. We're going to be moving into the question period within the next couple minutes or so. Just
yeah, I'm finishing now.
Okay, cool, awesome. Thanks. Sorry about that.
So future directions, we're working a lot on the mechanical characterizations of the materials because some of them we need to look at this carefully because of the applications. Also, hemocompatibility and thorough cytocompatibility assessments. We're working on that now in vitro, cell proliferation and differentiation, and in vivo tests, which we were hoping to start this year. Let's see if the pandemic lets us. So I want to thank you for your attention. Here are some of our collaborators from around the world. And if you have some questions, I'll be glad to entertain.
Awesome, thank you. I'm going to read off some of the questions that have been shared in the chat.
All right.
So our first question is, you mentioned that sometimes biomaterials have problems with their strength. Have you ever planned to use a mix of biomaterials and normal recycled plastics?
Right. No, we have we have thought about it, but we have not gone into that. We're focusing because there is such a great diversity on the sources of starches. We have found, for example, very different behaviors of starches from different sources. So we're focusing more on studying the behaviors of different starches or different pectins in terms of when we modify them, how that changes properties of the hydrogels and things like that. But it's interesting. I mean, we have thought about it, but there's only so much we can do at the moment.
All righty, sounds good. Our next question is, when talking about combining starch and the chitosan, you mentioned collaborating with a professor in food sciences. Could you talk more about the role of a collaboration in that research?
Yes. So when I started working, actually, there was the first time that I worked with starches, and I knew basically nothing about starches other than, it's an important energy source. So with this collaborator, we did a lot of the characterization of the starch modification per se. So I did learn a lot about the modification, the structure, the rheological properties, and we keep collaborating on that because every time you do a new modification, you need to see how the starch properties change. And many of the modifications we're using, they also have a lot of food science applications, especially for coatings, food coatings, and things like that.
All righty, cool.
I hope that answered the question.
No, yeah, sounds good. Thank you. So our next question is, since the material is readily available, what do you think is/will be the main obstacle preventing the use of agricultural waste in the biotech industry?
Right. So if we talk about a reality in Ecuador, I always say go local. The main obstacles that we have here is the actual infrastructure for taking this from the bench to the clinic. So that's the main obstacle that we have, and Ecuador is only starting now to work on these regulatory issues, especially bioethics and animal studies and things like that. This is by far the main obstacles that we face, and as we do research, we also have to be working with the agencies to find out a way to make this work.
All righty. Our next question, it's a follow-up question, not necessarily a follow-up, but it came in that same string. Do you think the popularization of this technology is likely to help agricultural and low-income communities, or is it rather going to cause a negative impact because of the new way the value of food is perceived and driven away from the elementary field?
Right. So one thing that's important to point out here is that we're not using food grade material. I mean, for example, when I talk about potato, I'm not focusing on the potato itself, but the peel. So the waste, if we talk about starches, we're talking about extracting starches from agri-waste, so not the main food value. So if I think it would actually help because it's giving more value to this waste. So it's like an extra income that the producers could have. How this would happen, we're not sure, but we're hoping it's doable.
Okay. Our next question is, can you compare MOFs and hydrogels in drug delivery, and which one of them is proper for stimuli response mechanisms?
So we haven't worked with stimuli response yet. However, there are some reports that you could work with some starch modifications where you can have pH sensitivity, which means, I mean, if the pH lowers, the polymeric network will open up a little bit more, and you could have an antibiotics there that if you have an infection, pH goes down, the network opens, and you deliver more antibiotics and fight the infection. We haven't, this is in our plans though. The same thing happens with Chitosan, but we haven't worked with that yet. No.
All righty, sounds good. And we have another question. What are some possible applications of the cacao husk sponge that you are studying, both in biomedical applications and others?
So we're still working with the biomedical applications now. We're getting ready to do some, we've done already some, but we're getting ready to do more cell studies, especially for differentiation studies. But also now we're working with the agricultural biotechnology laboratory here at the university in mobilizing fungi, especially fungi that have important roles in fighting plant diseases. So we've been working on that, and it's actually giving very interesting results. And we're also looking into the absorbent properties, especially they have shown preliminary good behavior on absorbing oil, for example. So we're looking into that too, but this is very new, still very new.
All righty. And our final question before we move on to our thanks will be, how does availability of different materials affect research into biomaterials, such as the different resources in Ecuador? And then are there examples of research into one type of material generating broader discoveries that could be applied to different materials?
Hello, can you hear me? Sorry, I lost connection for a bit.
No worries, no worries. We can hear you again. Did you hear the question?
Can you repeat it, please?
Yes, we'll do. So how does availability of different materials affect research into biomaterials, such as different resources in Ecuador? And then are there examples of research into one type of material generating broader discoveries that could be applied to different materials?
So I'm not sure I understand the second question. About the first question, so as I said, there's a great diversity. However, some fruits, for example, are more widely cultivated than others. For example, if you look at passion fruit, pectin from passion fruit has better properties in our opinion than orange fruit for our hydrogels. But then we would have to find a way so that passion fruit is probably more widely cultivated. However, you have to see that for these applications, you don't need that such large amounts as for other applications. If we talk about, for example, pectin hydrogels, we're talking about concentrations of 0.5 or 1%. It's not like you need a lot. So availability in our case is not such an issue because from what we've seen from statistics, there's a lot of production of these species here. So luckily, that's not a problem. Now, the second one, I'm not so sure of. With every material that we've been working on for hydrogels and also microparticles, which I didn't talk about today, we have started to see other applications, and we've been working with departments in for fashion, for example, which was very weird at first, but it's taken a huge role. Also in the agricultural biotechnology, as I mentioned, encapsulating microorganisms and polyphenols and things like that. So yeah, every time you're working with a new material, a huge window opens up for other applications.